Hi, I'm Dr. Natalie Marks, and I'm here with another Fountain Report conversation. I'm joined today by Dr. Eve Hanks. She is the CEO and founder of myRNA, an innovative diagnostics platform. Thanks so much for being here.
Thanks very much for having me.
So tell us a little bit more about your company, how you got started, and where did this idea come from?
That's a good question. And I think my background as a veterinary surgeon, I never thought for a minute that I was going to go into innovation or diagnostics or generally into science and research, actually. So for me, initially, I left university to go into practice, and I was working as a veterinarian on farm initially, then with horses, then in small animal work and a mixture of all. And after a while, I went into some emergency work as well and started to do some telehealth too. So I really saw like practice from all these different angles. I think that really helped to form more of an opinion about what I felt needed to change and what I could maybe do to make a bit of an impact in the industry. And I did a certificate in medicine and then a PhD in immunology. And really, after that, I felt I had some scientific expertise, the background, the pedigree, perhaps, to start thinking about innovation. So it was while I was working at a university in Scotland as a clinical pathologist that I started a science project to understand if microRNAs could be used to understand disease in a bit more detail, to give us a little bit more of an illumination of some of the disease pathways that exist. And my first, I suppose, thought with that was to see if they could be used as a diagnostic. So that was where this whole idea came from.
Could you go a little bit more into what microRNAs are? I mean, I think most of us are pretty familiar with classic RNA, but why is this piece of information just so telling in the diagnostic space?
Yeah, I think there are a few reasons that microRNAs are important. And actually, a lot of people may now have heard of microRNAs, particularly after last year, the Nobel Prize being given to the two scientists who first discovered microRNAs. So they are very new to science. And initially, like you say, we understood RNA in the scientific field, messenger RNA really well. But microRNAs are like the project managers of the immune system. So it's perhaps a little bit surprising that we hadn't stumbled upon them before we did. But part of that is because they have such a wide range of effects. It wasn't clear that there was one molecule doing that. So they can turn genes on and off. They can break down messenger RNA, so they change our protein output. But they also can act directly on molecular pathways. So they're really powerful. And they come from everywhere, I think is also a really interesting fact. So there's microRNAs in bacteria, in viruses, in plants, in all mammals that have been tested, including humans. And they're so important to us that they can, without microRNAs, life can't exist. So they can keep us healthy, but that also means they have the power to go wrong. And when they go wrong, we see things like cancer, we see a lot of chronic disease. So immune-mediated disease, heart disease, kidney disease, etc. But they also will control our inflammatory responses to other pathogens. So if we were attacked by a virus or have sepsis, for example, all of that is microRNA controlled. So they're really very important.
And so, as we talk about your platform, what are some of the unique features of it? And what, as a clinical veterinarian, how would we expect to interact with that platform?
There are a variety of ways that we currently, as veterinarians, use diagnostics, and we tend to use them in a binary form. So we tend to use diagnostics to say, is a disease there or is the disease not there? So we look in, I suppose, instead of asking further questions, we just have that approach of presence or absence of disease. And in fact, sometimes what we're doing is asking the presence of multiple diseases and more sophisticated diagnostics. But when you're looking at things like microRNAs, and I know it sounds a touch science fiction here, so it is a bit of a different way to look at diagnostics, you can actually interrogate those microRNAs as a panel of biomarkers. So a certain group of microRNAs will control the health of certain organs within the system. And you can actually line those guys up as a panel. You can test your sample and you can ask if disease is there or is health there. and you can ask what stage that health or disease is at. So they map the entire journey from wellness right through to the end of illness. So you can understand how the disease is progressing. You can start to ask questions about what might happen if you give a certain medication or you don't give that medication. And you can also start to separate out cohorts of patients, some who are going to advance quickly through different stages of a chronic disease, and others that might be stable and they don't need any great intervention from a veterinary surgeon at that point. So the interaction is quite different and it's driven by advanced modelling. So microRNAs are new, but advanced modelling isn't as new now. We see a lot of technology using artificial intelligence to decode a little bit the data we can get. So that is very relevant to the microRNA space as well, because as soon as you start to work with a panel of markers, and there could be 20 or 30 information points, then using advanced modelling is the best way to fit that pattern against a model that's already learnt what stage of disease or what progression of disease is ongoing.
So there's a lot there, right? And there's a lot of possibility and opportunity. Where do you, as the CEO, founder, and certainly the leader and driver of this platform, where do you see this having the most immediate impact? What species, what diseases, where are you heading with this, and what can we kind of look forward to in the future in both the small animal and large animal space?
You're right to mention both, actually. So we are species agnostic at myRNA for a few good reasons, because microRNAs have a common ancestor about 500 million years ago. So it means that all of us, humans and mammals in general, have microRNAs that are well conserved. So the microRNAs in my heart are the same as a horse's heart, the same as a dog's heart, the same as a cat's heart. So it's just really effective R&D from that point of view. So the work we're doing in small animals doesn't have to stay in small animals, and the work we do in farm also could potentially move over into other species. So we are open to that and we keep everything under review. But the two big impacts we'd like to have are that in the pet space, it's really important to spot early disease and then effectively manage it. So if we can tell that a dog has early heart disease or a cat has early kidney disease from literally the day it occurs, from the first time that we would consider it to be out of health, then that allows us to manage that patient, extend their health span, not just their lifespan. And as owners, that's what we're looking for. So I think therapeutic advances are important, but therapeutics only work if diagnostics exist to guide them. And so I think monitoring and early diagnosis is so important.
How are we detecting the microRNAs, and what are we anticipating this going forward as far as the ease for a veterinary practice or a farmer to be able to do these kinds of tests?
So microRNAs are actually really useful in this sense because they are found in a range of different body fluids. So you can find them in saliva, funnily enough, but more perhaps in terms of the research we're doing, the more obvious collection points are blood samples, urine samples, and milk samples from cows. So it remains a reference lab model, and that means that the vet or farmer under the veterinarian's direction would take the samples and send them into the lab for analysis. But because we can use things like urine, we can build in telehealth, for example. So we've got different streams and different ways that people can interact with vets to find out when to use a diagnostic. And also at-home testing sent into a lab allows perhaps cats with heart disease, dogs with problems that don't want to go in. They've got anxiety issues, for example, that don't want to go into the vet, still to be managed effectively. So that's something that we're very consciously building into the platform.
Thank you so much. Again, Dr. Eve Hanks, CEO and founder of myRNA, an innovative diagnostics platform that is here and ready to help us change some of our diagnostic strategies. I'm Dr. Natalie Marks, again here with another Fountain Report conversation. Join us next time. Thanks so much for listening.